You're going to hear ad nauseum about the tissue part of it. I know you had some questions before about what happens, but you're going to know more than you wanted to know probably. Okay, so journey of a brain tumor. In addition to the sort of the usual diagnostics, so I can answer stuff about what happens on the clinical side, what happens to your tumor after it's taken up by the neurosurgeon. But that's not the only thing that might happen to tissue. And what I wanted to do today, or actually what I was asked to do today, is to just give you an overview of what happens to all the little pieces, where do they go, you know, what's their fate. You know, here as patients, you see the neurosurgeon, and then you know, the neurosurgeon operates, and then some kind of time later, there's a big black box there. There's a report generated, right? That report might take a little while, probably longer than you'd like. So the other thing I wanted to explain is what are all those steps in between? Why does it take a week to get your report back? So a report is generated, and then that report goes to your oncologist, and they discuss the result with you. So those 3 question marks, I guess over there, that's all my stuff. I'm that black box. So hopefully by the end of today, what you'll see is that there are many steps in between to generate that diagnosis. There's a microscopic diagnosis, there's a molecular diagnosis, and those things are integrated into a final picture where we can classify the tumor. And then afterwards, I'll explain that. It goes into archives. And then there's another stream that's a research stream for materials, and that may or may not apply to you. But we'll go through each of these streams sequentially. So first of all, just for everybody who comes to neurosurgery, there is this, basically any piece of tissue that comes out of the operating room is seen by a pathologist. So that's the start of it. Here you have the OR, there's some team of neurosurgeons there, and that little orange-capped P cup is where your tissue ends up. So that little sterile cup, they will put the tissue in there, and they'll call a porter, and that porter will run it through the building over to pathology. In the pathology lab, so up at the top, you can see, oops. So there's the surgeon. They are going to send that tissue to the gross room. So I don't know if you guys have heard of this terminology, but right into the mic. Okay. When it gets sent to pathology, it ends up in a place that we call the grossing room or the frozen section room. And this is a picture of what that looks like. Here you have a pathologist and probably a trainee on the left, and they are receiving that little vial of tissue. They're opening up the vial and they're looking at it. They're seeing how big is it? What does it look like? Certain tumors actually, again, this is probably more than you want to know. Certain tumors look more like firm, tofu. Other tumors look more like jelly. And each of these little characteristics, that kind of guides us to say, you know what, that part of the tissue right there, that's what I want to examine under the microscope, because that's the money piece. And what you want to do is you want to make sure your goal when the patient is on the table, neurosurgeons, you know, the head's opened up, and they're waiting for you, is to tell them, yes, you have diagnostic tissue, or no, you don't have diagnostic tissue. You need to go get some more because, you know, it's not enough. So that's what they're looking for over there, right on the left. And then the next panel shows what we do to not just look at it with our eyes, but we make something called a quick section. This is a frozen section. We quickly freeze the tissue, and we cut it on that machine. That's the second panel. That's called a cryostat. We freeze the tissue. We cut something very thin. That you see here, there's a piece of tissue, and it's in some mounting media. That gets mounted on a slide very rapidly, and then we look at that tissue under the microscope while the patient's on the table. So this kind of turnaround time for an intraoperative preliminary diagnosis is about 20 to 30 minutes. So that's while you're in the gross room. That preliminary diagnosis, we don't have everything, right? So this is one thing I want to, getting to your question previously about, you know, the different steps of information and why it could change over time. Even when we have pathology, you know, our tissue is in our hands, we're looking at it now. These rapid diagnoses that you get in the frozen section room as a prelim, they're based on frozen tissue. We don't have all our stains. We don't have any molecular information. We can get about 90% there. We can say, yes, it's a glioma. Yes, it looks high grade. But maybe we can't say, you know what, it's an anaplastic oligo versus an anaplastic astrocytoma. So we can't go all the way in the frozen section room. But we do get a preliminary diagnosis. We also get enough information to figure out what do we need to do with that tissue if we need to do something very special. Do we need to, based on what we see at first, send it for molecular studies? Do we need to send it for tissue culture? Do we have to send it for this or that thing? So that's a triage point. Actually, there are some points where, based on what we see, it is not what we expect. So on the imaging, that it looked like a certain kind of tumor, and actually now that we have it and we make that quick diagnosis, it's something else. And that the most critical role is to guide the surgeon. So there are some tumor types that are not treated by resection. So if I say, you know what, this is a lymphoma, they're going to stop, right? They're not going to try to resect that thing. They're going to give it chemotherapy and it will respond. Alternatively, occasionally we've seen things where it's not a cancer at all. It's an abscess. It's an infection, right? And it looks on imaging like a tumor. So they might do something very different in that case. So the whole role of this is to guide what happens right now. The next phase of this is to generate what we call permanent sections, which is our routine looking at slides that are prepared in a sort of longer processing time but yields you better images kind of thing. So that clinical processing, it involves a couple of things. What you see on the top left is embedding the tissue, once it's fixed in formalin, embedding it into a wax block, and that wax block is called a paraffin block, allows us to cut better sections on a different kind of, you can see that on the second. We're cutting very, very fine tissue sections, and we're going to mount those on slides. Tissue has to fix, and then process overnight, and then the first slides come out the next day. So already you're talking maybe 2 days after your procedure before we even see any good slides. At that point, we can view it under the microscope and say, okay, what are the next quick tests that I can do? So some tumors, at the point where we see the first slides, we know what it is, and we can sign the whole thing out, and it's very clear. Other tumor types require some special studies, immunohistochemistry, antibody studies to detect certain proteins on there to help us classify. If you need those kind of tests, that takes a few more days, so each test takes overnight. So on the top right, you can see a technician loading an auto stainer for immunostains. These are special antibody stains to detect particular proteins. And then finally, when you have all of that information, this is a multi-headed microscope. You can see somebody looking at the microscope and then other people looking at the same time at the same image to decide, okay, here are the features. And then we can classify the tumor. So at that point, you're talking maybe, let's say 4 to 5 working days after your procedure. This is kind of stuff we see under the microscope on our routine H&E slide. And what I can say is that they all look kind of different, right? And this is what we're trained to recognize. We're trained to recognize that on the top left, that's a WHO grade 1 pilocytic astrocytoma. If you have one of those and you have it completely resected, it will never come back, right? Or depending on where it's located. But if you get the whole thing out, 99% of them never come back, right? That's not going to be your issue. That looks very similar, but it's importantly different from the next one, which is WHO grade tumor, which will infiltrate through tissue. There's a relatively long survival, but it's not like a WHO grade 1. And now as you move to the top right, that's a glioblastoma. So these are all astrocytic tumors. Our eyes as neuropathologists are trained to recognize the special features of the cells, how dense the cells are, what the nuclear shape is, what proteins they express in order to classify these. And then there's some other ones on the bottom. It's just to really impress you with what I do every day. But anyway, this is the brunt of making our diagnosis. Most of it is on the H&E, and then once we have narrowed down this kind of classification, we run extra studies. This is our, I suppose, diagnostic Bible, as you say, is a WHO classification. Everybody gets a WHO classification. There's a reason for that, is that no matter where you're treated, no matter where you're diagnosed, there's a standard in the world that says, okay, they called it this over there, I'm calling it this over here, and there are treatment protocols written on how you classify the tumors. So one of the things that's all just without many special studies. One of the things that we've recognized over the last few years with that technology like whole genome sequencing is that things that look the same are not always the same. So, before I showed you these beautiful images, they're pink and they're blue, but that's something that you see by your eye. Now what I'm showing you is that on the left, that's something that every single pathologist in the world would have called a GBM, a glioblastoma. It's got vascular proliferation, it's got necrosis, it's astrocytic cells. But if you study a lot of those, what you'll find is that there are subgroups within there that are molecularly different. And when I mean molecularly different, they carry different mutations, they express different proteins, they express different RNA. And among those different subgroups, there are some, I would say, fairly big differences in prognosis for some of the subgroups versus other of the subgroups. So in some cases, for some tumors where we know this information and can act on the information, we will run additional diagnostic tests. Those diagnostic tests, I won't go through that one. Those kind of diagnostic tests are actually looking at the DNA level itself and the chromosomes themselves to count chromosome numbers, to look for translocations, to look for mutations in particular genes. So on the left, you can see that's a cytogenetics study. The screen is showing a chromosome spread from a tumor, and that means that little blob of little white things on the screen, those are all the chromosomes that came out of a single tumor cell. And in the case of gliomas, if you carry a certain kind of alteration called a 1p19q co-deletion, that puts you in a very specific subgroup of tumors of oligodendrogliomas, and they do very well. But it could look the same as something that doesn't carry that. So that's an example of cytogenetics. And then we also do molecular diagnostics, which is looking for the mutations themselves, not on a large-scale chromosomal level, but on an individual base level at the DNA level. So this information we call ancillary testing. Most of the time it's ancillary because the main classification was already done on the stains that the tests that I talked about before. This would give you added information that within, let's say, diffuse gliomas, that you are a specific type with a specific prognosis. It may give you information on the likelihood of whether you'll respond to a certain therapy or not. But it doesn't change the WHO classification for you. Sometimes we do have trouble recognizing just on the sort of the pink and blue type of images what type of tumor it is. And there are certain tests that are special molecular diagnostics tests that will help us classify. That's in a smaller percentage of tumors. So most cases, by this time, most cases can be signed up within a week. 5 to 7 business days of the surgery. If you need these kinds of ancillary molecular tests before your treatment starts, for example, these type of things take perhaps a week and a half more. But again, they don't affect the overall grading. They might affect something else, like whether or not you qualify for a certain study, what the overall prognosis is, even though we can't change the therapy. That's why it's ancillary. And then finally, we would take all that information, the initial information, plus the molecular information, and it would be up to the neuropathologist or the pathologist to integrate all of that information into a final diagnostic report. That final comes in when all the information is in. Often it doesn't change the grade, it doesn't change the overall classification, but it has all the little bells and whistles on it. Now, I will say one thing about pathology and neuropathology is that it does require specialty training in neuropathology to recognize these tumors. So, anatomic pathology is one general area where you might look at lung and colon and all these other organs. Neuropathology is a very special area in pathology where you need specific Royal College training. And there are only about 40 neuropathologists boarded in Canada. We have 4 neuropathologists in Calgary alone, so we're doing pretty well in terms of neuropath. But one thing is that I don't know if it applies to any of you, but sometimes tumors are diagnosed outside of a major center. So they might be diagnosed in a peripheral hospital, a community hospital where there's an emergency procedure, And sometimes it might be beneficial to have that material reviewed by a boarded specialist neuropathologist because we have training in that particular area. So then what happens afterwards? So there was this little piece, remember that paraffin block I showed you, that little wax block, and they're cutting the sections, and what happens afterwards? Well, it goes into archives. And this is stuff that I think some people don't know, or you kind of wonder, well, what happened to it afterwards? The slides, the diagnostic slides, are kept in pathology, in the pathology department. They're kept on site for a couple of years, and then we move them to off-site storage just for space reasons, but they're kept in archives. The same thing with the paraffin block. That thing is retained and kept in archives. So on the left, you can see some slides. On the right, some guy, I don't know, I stole that from the internet. Some guy, but what he's doing is he's in the paraffin archives. The reason you want to know this is that that paraffin archive is good for actually a couple of reasons. Number one, you can go back to it, actually years and years later, and it's still preserved. So we've had that happen where a child was diagnosed with a certain tumor, and it's like, well, that wasn't really behaving, he's done great, 10 years later. And then you can go back and you can run tests because, again, science has advanced. You now have these tests, and you're like, okay, it was actually this, and now we would actually manage this way. So it's something that we can always go back to. It's also another way to contribute to research, and I'll get back to research afterwards. So how long are these things kept? There are intra-hospital differences between how long material is kept. There are guidelines that are suggested from various bodies of how long is the minimum that they suggest hospitals retain tissue in archives. It also varies country by country. It varies by whether or not you have a public healthcare system versus a private healthcare system. Sometimes for-profit hospitals don't retain tissue for as long as they meet the minimum, and then they get rid of it because it costs them money to store the material. Some, basically, Children's Hospital, Alberta Children's Hospital, they will keep material forever. It would be great if we could keep material forever, but sometimes it's not feasible. There are minimums. So generally, that type of minimum hovers around the 20-year mark. So your material will be there for 20 years, next couple of decades, and sometimes indefinitely. I talked about archives being a resource for second looks, but second looks can also be important when it's not just in archives. People have asked me before, like, oh, should every case get reviewed, if I'm coming from somewhere else? So there are certain practices that we have in place, at least here, and that are recommended. So at least in our practice in Calgary, all the malignant diagnoses, they get a second neuropathologist. We have a consensus conference every Monday where all the slides are reviewed from all the malignant cases. So basically it takes two people to say, yes, I agree, and then we finalize the report. So like by definition, it's had two sets of eyes already. But there are other special situations where a second pair of eyes is nice to have. I mentioned before, when there's a patient who's coming from another hospital and is going to be treated here, or they're coming from another country and they're going to be treated here, we would like to review the material from that surgery that they had elsewhere to make sure we agree this is how we would classify it here, and we may run molecular diagnostics tests here that they don't have in the outside hospital. So just for standardization, that's another time that we have second looks. Occasionally, very rarely, there are cases where we don't know what it is. And then we might send an external consult to, let's say, somebody who wrote the book, or like has seen the only 5 cases of this rare tumor type and is located at Johns Hopkins. So we'll write a consult letter and say, hey, we think it might be one of these things, but we've never seen it because there are only 5 cases in the world. Do you think that's what it is? So that can take some time. If you get to that point, which is quite rare, it may take a month to get an opinion back because then they have to run all their tests and ask for our material and confirm the diagnosis. But in general, if you're having doubts, I would say, about your diagnosis, you could always talk to your oncologist and say, you know what, I don't really understand my report. Can you explain it to me? Is this something that sounds pretty routine, that everybody has this, or does it sound like we need to look at the case again? I don't think anyone is offended by that. Everybody really, if you have questions, deserves a second, at least discussion. Okay, somehow lost my clicker. Okay, so that's what happens on the clinical side. We do the microscopy, we do the molecular genetics, we integrate that information to generate the report, and then material goes into archives. What I'm going to talk about now is something that is sort of optional, but is where a lot of tissue does end up out of the altruism of patients and their families. And that's a research stream. So at the point where, if you go back to that grossing room, right, where everybody's huddled around looking at the piece of thing that looked either like tofu or jelly, the pathologist made a decision. They said, you know what, I have enough diagnostic tissue here to classify. I think it's this thing, I am fairly certain. And anything that is in excess of what is actually required to make a diagnosis has the potential to be used for research. It's just extra stuff, right? You don't need it. You've already gotten everything you need to make the diagnosis and to run your special studies. What can happen then is that you can donate that tissue to tissue banking and research. And that involves informed consent, right? Because this is not part of clinical care anymore. Clinical care took the tumor out, but instead of throwing it in the excess into the trash, what you're doing is you're giving permission for potential research to be done on that tissue. And that can include a lot of different things, and you can sort of say, well, I don't want to do this, or I don't want to do that. But in general, you are consenting to have the tumor bank or the researcher access your medical records to see what your diagnosis is and what your follow-up was and what the eventual outcome was. So to be able to go back to medical records, to collect the tissue, to collect it and freeze it and treat it in a way that it can be used best for research in the future. It might consent for cell culture, and that's something that I'll explain that we do very special here in Calgary, is we have a lot of capacity for cell culture and molecular studies, the rest. And a couple things to note about consenting for research. I won't go through all of ethics and consent, but it is entirely voluntary. Patients can consent or they don't have to consent, and there's nothing that affects your clinical care whether or not your tissue is donated for research. The other thing that we do is we protect your identity. So let's say the tumor bank might have your information, but when they distribute that tissue to external researchers, if the external researcher does not have permission to contact individual patients and only has permission to use de-identified information, it just gets a number and it's anonymous, right? So they can't track it back to you. The last thing I don't have up here has to do with it being entirely voluntary, is that you might consent to use your tissue for research, and you also have the right to withdraw that consent later. So if you somehow decide, let's say, you know what, I don't want my tissue in some freezer, for the next couple decades, you can say, you know what, I withdraw my consent, and that tissue will be destroyed. So anyway, the first step in tissue going for research is the consent. This is something that, so I run the tumor bank. I'm a neuropathologist, but I also run the research tumor bank for the brain tumors in Calgary. For every patient who consents to donate tissue to research, we send a collection kit. A collection kit will have a couple more of those orange B cups for things like tissue and urine. It will have blood draw tubes. So this gets to what is required for biomarker studies. I forgot who you asked about biomarkers. The way that Jay Esau and others could run their studies was that patients had consented for research. Even though they didn't know what the research was, we had collected blood on all of these tumor patients, and we had collected serum and plasma and white blood cells beforehand, and that was banked. And so at the point where somebody had a good question, that stuff was in the bank. So when we collect tissue, we try to see, okay, what are all the things that could be done in the future that I want to maximize now when patients are consenting for research, because we don't know what it might be for. So there are some barcoded tubes there, and then that kit would be, the tissue's getting triaged in the gross room, and then it ends up with a neuropathologist or a tumor banker or a tumor bank technician or whoever is running your tumor bank. So that's me in my lab, and that's where the tumor bank is. You can see some boxes in the background of things that we use to prepare the tissue. On the right, you can see, okay, well, we have these racks. We aliquot tissue into vials. All those vials, there are 96 little vials in each of those on the bottom left, in each of those racks. And if you turn that rack on its side or look at the bottom of it, underneath every single tube is a barcode. This is how we keep track of our tissue. It's all barcoded. And it doesn't have your name on the side, right? So if the whole, if the box gets stolen, if it drops on the floor, you don't know what it is, you just put it back in the box and you scan it and all your information is back. And then if if nothing special is being done with it, it will get annotated in terms of diagnosis and all these other things, tumor content, and then it will be put into storage. These are liquid nitrogen storage tanks where we keep our frozen tissue until it is needed for a research study. In terms of our biobanking in Calgary, we're not the biggest city in the world, but our biobanking is well developed in that we do quite sophisticated things with the tissue when we receive it from patients. So many banks will collect tissue and then they freeze tissue, right? And then it gets used for future study. What we like to do is not only do that, but we would like to preserve our cells and our materials in a way that it's not just a frozen piece of tissue that can never be used for anything. You can grind it up and you can look at the DNA, you can look at the RNA, but you can't do anything alive with it. I mean, it's sort of like the steak in your freezer. You can cook it, but you can't grow a cow out of it. You can't study cows alive. So what we would do is we would try to treat our tissue in a way where we can use it for live experiments. And that means cryoprotecting it in a certain way. It also means putting it into culture. So we make a special media, it's like a soup with special ingredients, with growth factors and certain salts so that we can try to grow those cells in the lab. And then we can freeze down aliquots of those growing cells. Those cells can later be used for making animal models so that you can study. They can be used for in vitro drug screens for experimental studies. So this is what our bank does. Here's a picture of somebody in my lab who has this is what would happen. He has a pipetter there and he has some media in front of him. And so he's taking tissue from a patient and he's starting cultures. So he would grow those cells in a dish and then split them and make sure that they are identical to the patient's cells that came out of the patient. So why do we do this? There are lots of cell lines out there. We do this because there are cell lines out there and they've been around, in the scientific community for decades. One of the things that we found in the past, I would say 5 to 10 years, is that the way you culture cells can really make a difference of what they behave like. And so the way we used to culture cells, let's say 10 years ago or 20 years ago, it changed what they were like. So the glioma models that you have from ATCC or any of these other big companies, they don't invade brain tissue. And that's one of the critical things about gliomas is they invade tissue. If you want to study invasion, you're not going to be studying the right thing using those old kind of cell lines. So these, the way we do it now, they recapitulate many of the features of the tumors in the patients. They invade, they have angiogenesis, they have inflammation. So they're really quite a bit better in terms of model systems for studying particular cancers. On the left, you can see that difference in invasion. So that's BTIC stands for brain tumor initiating cells. So these are cells we grow. So you might have also heard of brain tumor stem cells. That's another term for them. On the left, you can see that tumor, it's kind of like, it's if you had a handful of marbles and you threw them across the room, they kind of spread out and they're going to roll under the table and in between and kind of diffusely infiltrate this room, right? That's what gliomas in patients look like. That's how they invade. The serum line and the traditional glioma cell line, you see a difference in the way those cells are behaving. It's like a little ball, right? And that really doesn't reflect very well what goes on in patients. So if you were a scientist and you wanted to study gliomas, you would prefer to study the things that look like BTICs. So we're generating a whole series of BTIC lines whenever we can. Culture cells, we do. We try that for every high-grade glioma and every high-grade pediatric tumor that comes across our bench. And then I'll just segue a little bit. I don't know how I'm doing for time. My bad. Okay, I'm going to keep going. So it brings me to one of the large program projects that we're involved in that's based on this human-derived cell, these human-derived model systems. Calgary is actually the lead center in a multi-institutional project that's sponsored by the Terry Fox Research Institute that I guess the long title is Modeling and Therapeutic Targeting of the Clinical and Genetic Diversity of Glioblastoma, but basically the short title would be Therapeutic Targeting of Glioblastoma. This is a disease that hasn't really had a lot of progress ever, for a long, long time. So we want to focus on these high-grade gliomas. And again, we're generating these new BTIC cell lines from all the patients who consent to donate tissue for research who have glioblastomas. The premise of this is that we are going to have enough cell lines to model the diversity of GBM. As I said, GBM has different subtypes. Has preneural subtype, has mesenchymal subtype, has classical subtype. So when you study them, you want to be able to say, you know what, I'm studying this type, or this type, or this type. Or the drugs that I put into screening studies, only one type responds to that drug. Or is it all types? So this is why it's important to have numbers. So far, in terms of classifying and sequencing these, we're going to do whole genome sequence, or we are whole genome sequencing all of these lines that we generate. So far, we've done over 50 at the DNA and RNA level. And we can see that our methods are generally pretty good for culturing and maintaining the genetic profile of these cells. On, I guess, the left, I'm so terrible here. On your right is the tumor tissue that was received, and on the left is the cell line that we grew from that sample. And this circus plot, it's a circle plot, and it represents all the chromosomes in the genome, shows areas of chromosomal gain and chromosomal loss in the tumor. And just at a very low, basic level, standing far away, you can see, they're very similar. So the things that we're growing in the lab are reflecting what's coming out of patients. And we do have representation across different subtypes now. So in terms of the giant scheme of the project, it would start out with the patient samples. Everything we do is patient-oriented in this particular project. And then we have a high-throughput screening core in Toronto that's screening thousands of compounds against those cell lines that we generate. And we also have a sequencing core in Vancouver that's doing incredibly detailed characterization of the cell lines, so that when there is a drug that seems to work for several of the cell lines, we can ask, is there something associated with that response? And then we have a preclinical core back in Calgary again, where once we have hits from the screening, we can validate those hits and then move the studies on to further sophisticated studies, for example, in animals. We also have a clinical trials core eventually that will carry that potentially promising drug into clinical trial. Thank you. One of the things that we do is we can screen for libraries that exist of compounds where, let's say, drugs have been developed for other diseases. Pharma companies have been developing compounds forever, right? And then there are a bunch of natural compounds out there. So there are libraries, that means whole sets of thousands of these things that we can use in high throughput screens, that's HTS. But we're also a mechanism for if a drug company, we're actually quite high functioning now because we've got sort of the soup to nuts workflow here, that drug companies have started approaching our group to say, actually, can you test our drug in your pipeline? And so we do have a number of external compounds that are coming into the pipeline as well. I won't do that. So yes, we've frozen GBMs banked since we started collecting is in the hundreds, almost 300. Banked since the TFRI, Terry Fox project started, is almost 100. And then we've generated more than 100 lines now. This project started 2.5, almost 3 years ago, but that's a lot of progress within 3 years. Again, we've sequenced already about half of them quite thoroughly, and we've already done quite a few high-throughput screens. In terms of our high-throughput screens, drugs have been screened against 23 of those cell lines, and that's more than 1,500 compounds against the 23 lines, so that's a lot of tests. We have certain libraries that we screen against, and we kind of have a workflow to shortlist. Okay, here's if you make this kind of sensitivity, you go on to the next stage. If you make this kind of sensitivity, you go on to the next stage. And right now, from the first 3 years of the project, I apologize, one of them is from a drug company, so I can't actually put the name there. But anyway, there have been 3 that have been identified that we're starting to write the protocol for a clinical trial, which would hopefully be starting in the next 2 or 3 years. So that's the culmination of this type of work. The other way to use that sort of information, because now we have all this giant sequencing information, we have DNA at every single base in the genome for these cell lines. one thing you can do them is you can just screen the cells against your drugs. But the other thing is you can take the information and you can try to predict, oh, based on this pattern of mutation, I think it's centering, I think the growth is being driven by a certain signaling pathway. And that's a different kind of analysis, a very, very computationally intensive analysis, but we're trying to see if this works. So we're taking structural variants, copy number alterations, point mutations, gene expression, and integrating that data to try to hone in on certain pathways. And again, from that kind of approach, we've identified a few that aren't close to clinical trials, but will be now taken to the point where we will like to perform some animal studies to see if there's any in vivo effect. Okay, and then I did want to finish. I just talked about glioblastoma, and I just talked about hundreds of patients and numbers and what we're doing on a large scale, and all of that research part. Please don't forget the clinical part, because that was kind of a long time ago, but at least for the research part, sometimes I know it can feel like, because we have hundreds, that a patient is a number, right? Or why should I donate my tumor because 279 other patients donated their tumor? But there are very good reasons for this. First of all, we don't know up front. When we're in that gross room, we don't know when it's a special tumor, right? It kind of look the same. It's a malignant tumor, we're going to bank it, we're going to try to grow it. You don't always know up front that you've got something very rare. And so I think it's really great when every single patient who has the opportunity, and again, it's totally voluntary, but to say, you know what, my tissue, maybe it won't help me, maybe it will help me, but maybe it can be used to help others, right? Maybe it can be used to propel research forward. So I wanted to transition to this particular cell line. It's cell line, it's called BT183AB, so it sounds kind of just letters and numbers. But this came from a patient, right? The parents of that patient are sitting in the back. That's the Browns, and you can see a magenta shirt, a blue hair, and you can see them in the picture up here with their son, who was 2 years old when he was diagnosed with this very aggressive, very rare pediatric tumor, one that we don't have any treatments for. Alexander ended up passing away from his tumor. But I wanted to emphasize the impact of donating to research because they consented to this. There was no way to study this tumor in the lab because it's so rare that a cell line did not exist. There was no tool, to look at it. There were various labs that had collected frozen bits and only recognized afterwards it was a special tumor. But up front, we didn't have that resource where somebody got it, they cultured it, and now we have a resource to study in the lab and do experiments. So they consented for research, we got this crazy thing, and in fact, we thought it was another common tumor type. We thought it was a medulloblastoma, because that's what's common in kids in the cerebellum. We don't ever think it's going to be one of these. So we ended up generating a cell line, and just to compare, it looks exactly, when you put the cells into a mouse, it looks exactly like a patient tumor. So you can see Alexander's tumor, and you can see the mouse tumor right next to each other, and you really can't tell the difference. They carry the same genetic aberrations. They carry the same protein profile. So now, because of one patient's donation, there's a field of research that has opened up, right? That's from one family. And again, you can't tell up front whether you're going to be that rare one or not. And every single sample helps in this way. So in terms of this particular cell line, I should update the little flags there, but because there was nothing there, as soon as we said, you know what, we've got this resource, that resource has now been sent at the request of investigators in other countries. Well, first of all, in Canada, it's been sent to Vancouver, it's been sent to Toronto, it's been sent to Montreal, it's been sent to centers in the States to investigate, it's been sent to Switzerland and multiple centers in Germany who have research programs along the lines of rare pediatric tumors. So it's kind of incredible, and I'll have to say, not everybody can have tracking like this. I know because it's such a rare thing and people request it all the time, I know, exactly which studies it's in. For the most part, we don't track that. We could say, okay, a person who donated glioblastoma, you have contributed to this kind of study, but I can't list all the different kinds of studies. But just from that one sample that was derived in 2012, this is what's come out of it, right? We're not at the stage where we can screen drugs. We don't even know anything about this tumor, but what it's done is it's enabled research into the basic biology of this tumor type. Here, I mean, I don't know if any scientists in there, but Nature Genetics is a very well-regarded journal where new findings are reported. So that was supported by this sample. Different classifications of pediatric tumors, that was supported by this sample. And then that's our report of the cell line itself. So that's the power of donating to research. So in summary, I'm going to summarize. I don't want you to walk up here. That's my fear. So in summary, this is where we started, right? We started with a patient. There was a tumor. You went to the OR. There was this big black box, and then the report got spit out. And then what I hope I've kind of laid out the framework of where all that tissue goes, where it ends up. And also the other thing I didn't say, after our research studies that we're doing actively, it is banked, right? Excess stuff is banked so that in 10 years, when somebody has a new question, there's material there for them to go address those questions. So that's the whole scheme of it. Hopefully I've answered, the journey of the tissue, at least, of the brain tumor. And I'd be happy to take any questions. I'll have to say, I have to thank lots of people. I mentioned I showed figures and so on and so forth. The Brain Tumor Bank in Calgary is run by lots of different people, neurosurgeons, nurses who consent, technicians. Most of all, really, we wouldn't have a bank if patients don't donate to the bank, so thank you, patients. My lab does a lot of research around those areas, and then we have lots of collaborators locally, nationally, and internationally. At the bottom, I've put some of the funding agencies that my lab and actually our research group in brain tumor has received to support this type of work. Thank you.